So the next talk is Raj. There's coming down. Who's going to tell us about long-range energy transfer and a very tongue-twisting word, biomimetic. Got it right, good.
Okay. Yeah. All right, so thanks to the organizing committee for putting together a great program for today and the other great speakers, and for giving me the opportunity to share some of the work that we've been doing on OE in sort of some very exciting work that I think in sort of imaging energy transport in real time at the nanoscale. So we all know that plants are very effective at harnessing energy from the sun and transferring that to a reaction center very rapidly and very efficiently, such that that light energy can be converted into chemical energy for metabolism and for energy storage purposes. And as scientists, we are really interested in being able to replicate that process, not only for sustainable energy applications, but for really understanding how we can control light at the nanoscale. So can we make sort of molecular circuits where we sort of excite something and use that to make a nanoscale computer? Or can we understand how chemical reactions take place? And so in a biological system, what we have is we have this sort of light harvesting antenna called a chlorosome, which is an array of dye molecules which are packed in a very specific geometric arrangement. So this is shown for the green sulfur bacteria, where they assemble into these sort of tubular type arrangements. And these complexes of dye molecules capture your sunlight and with very high efficiency transport it to the reaction center. And this sort of capture of sunlight and energy is sort of the reaction that could be argued that sort of underlines the whole biosphere. So it's a really important thing for us to understand and study. But there are really two major challenges associated with understanding this and replicating this process. Firstly, on the materials point of view, we face a significant challenge. Plants don't really like being exposed to huge amounts of sunlight. If you take the chlorosomes outside of the plant, they will die. They require their protein scaffold, so sort of these big complex groups for them to survive. And if we're going to develop a material that's able to replicate this process, we need it to be highly sustainable and highly reproducible. You know, when it comes to winter, plants die, so we don't want our material to fall apart when it gets a little bit cold. And secondly, and almost equally, or if not more importantly, if we're going to be able to replicate this process, we need to understand exactly how it happens. And here the real challenge is that these energy transfer processes are taking place on the nanoscale. So they're much smaller than what we can normally observe with typical light-based techniques. They're much well below sort of the diffraction limit of light. And not only are they very small, but they happen on an incredibly fast timescale, so hundreds of femtoseconds, so 10 to the minus 15 seconds. So we have two real major challenges there. So on the materials point of view, chemists have made big inroads into developing systems that we can use as light harvesting mimics. So they've basically taken dyes, which these are three examples, that when under judicious conditions can be made to pack together and self-assemble into sort of different structures. So this dye, for instance, packs in all these dyes pack in sort of a brickwork arrangement, and depending on the arrangement of these sort of nanoscale bricks with respect to each other, they can self-assemble into these sort of supramolecular type structures. And so this is a really big improvement because these are photostable and we can put them in devices. So this challenge to some extent has been tackled. And so one of the systems I'm going to be talking about and present a little bit of data for in the next sort of couple of slides will be systems based on this dye molecule, which under correct conditions can be sort of self-assembled into these nice fibrillar patterns, which are hopefully good mimics for light harvesting complexes and may possibly be able to be used in sort of solar devices or nanoscale circuitry for harnessing energy. So one of the second challenges that I mentioned, and arguably a more important thing to be able to do, is to be able to actually image these nanoscale objects. And this problem has been tackled for many years by the biological community using super resolution microscopy techniques. So as I mentioned, the light visible light has a limit to how small an object that you can observe. So this is set as the diffraction limit, and it's typically half the wavelength of your light. So if you have a 500 nanometer imaging, if you're using 500 nanometer light to image something, you can only see something down to 250 nanometers. But what we want to see are things at the sort of 10s and 20 nanometer scale. And so what biologists came up with, and this won the Nobel Prize in 2014, is a technique of super resolution microscopy, and here's an example. So this is a STED microscopy. So basically we have one beam, two diffraction limited beams basically that we use to image our sample. One beam is the shape of a circle, and the other is a donut shape. And what these two beams do when they add it together is that one beam excites your sample, and the second beam basically removes the emission away from your excitation spot. So it basically turns off some of your emission. And this basically, by turning off the process, you are able to actually resolve a smaller number of structures and actually be able to see below this visible light limit. And as I'll show, the technique that we've been working on and developed effectively works on the same principle, sort of turning things off. So there's a final component, of course, to this transfer, is that energy transfer is a dynamic process. It occurs on a fast time scale, and so imaging alone doesn't allow us to see this. We need to be able to track this motion in real time. And so the femtosecond laser community has, for a long time, been able to use femtosecond laser pulses to sort of track motion of carriers over a specific time. So this is the pump probe technique, which has been around for many years. So one laser beam excites your sample, and then by delaying a second beam which interrogates your sample, we can build up a trace of what happens following photoexcitation. So you end up with some sort of kinetic, and from this kinetic, you can infer different things about your system. So having got this whole toolkit together, our group in Cambridge basically said, okay, can we combine all these techniques? Can we combine the super resolution imaging with ultra-fast spectroscopy? And that's what we did. So we built a pump probe setup. We compressed the light beams down to sort of 10 femtoseconds so that we could have very short light pulses to see what was going on. And then we put them through an objective. So that in this case, rather than just looking at sort of a kinetic, we actually achieved a spatial signature of our pump probe map. So how does this work in a bit more detail? So basically, if we imagine we have our sort of nanoscale wire here, the green spot basically is our excitation beam, and the red spot is our probe beam. So when there's no time delay between the excitation beam and the probe beam, we just see excitation of our sample. But if some energy, say, were to migrate from this excitation region to somewhere outside of it, as we delay our imaging beam, so our probe beam, this red, we should see a change in the spatial pump probe signature. So it's basically this shaded blue region. And so if we fit the spatial signal as a function of time, we can basically see at 20 femtoseconds we end up with this spatial extent, at 30 femtoseconds we might end up with this spatial profile. And by subtracting these two spatial profiles and building up a histogram, we are actually able to resolve spatial changes that are well below the diffraction limit. So basically our limit is set by how good a signal to noise we can achieve. And so typically in the experiments I'm about to show, we sort of can achieve sort of spatial motion down to the tens of nanometers, basically, 5 to 10 nanometers. So here's an example of sort of some of the sort of data we collect. So here is a big bundle of molecular aggregates, which is sort of imaged on just a diffraction-limited microscope. This is what they look like inside there. So you've got thousands and thousands of wires. And so what we do is we place our pump beam at this end of the molecular aggregate. And so what you want to focus on is sort of these blue spots, which is basically your pump probe signal. And so what we see when there's no delay between the pump and the probe, we just have our excitation spot. So exactly what we'd expect. And we can measure the size of it at time 0. It's 350 nanometers, which makes sense if we're exciting the 700 nanometer light. But as we delay the probe beam, so as we watch, so we delay the imaging after a certain time, what we see is that this initial sort of very small blue spot moves across the sample and increases in size. So our pump beam is still staying this small, it's still this size, so we're not adding any more excitation in. But what has actually happened is that excitation has moved from this end of the aggregate to this end. As you can see, the spatial pump probe signal has moved, and there's a change. And so what we can do is we can fit a spatial pump probe signal as with some Gaussians, and by subtracting the two Gaussians, we can see that the energy is actually moved, the excitation has moved sort of several hundred nanometers in a timescale of 800 femtoseconds or so. And so this is not just limited to one set of molecular aggregates. We tried another set just to check this might actually be a real result. So we took this aggregate, which looks like these sort of sheet-like structures inside there, and we basically placed our beam at the top of here. And at time 0, we see exactly what we expect to see, basically an excitation spot. But as we delay this imaging beam, this probe beam, with respect to time, we see the pump probe signal move spatially down the aggregate. And so I just want to, so similar things we're seeing here. And then we can fit it, and we get out our spatial profile. So just to conclude, so ultrafast microscopy is a new technique that has been developed that can image a wide range of quasi-particles, so excitons, plasmons, polaritons, condensates, etc. It's particularly suited to studying materials as a function of both dimensionality and in terms of how material structure. So we're not just limited to looking at organic materials, inorganic, hybrid, organic, inorganic materials are also very accessible by this technique. And unlike fluorescence-based techniques, we don't actually require on the molecule to emit. Because it's based on the differential absorption, we don't actually require an emission from the molecule, so we can look at dark molecules as well. This is, to our knowledge, the first time exciton motion has been actually imaged in a biomimetic system. And so it's the first proof for actually that these plants and bacterial systems can actually transport energy over long distances. And what we find is that the transport is unprecedentedly fast, so it's about 100 times faster than what we would expect or theory has shown. And it's long range, it's sort of 400 nanometers. So with that, I'd like to thank basically our collaborators in Oxford, the Kokura group, who have worked with us throughout development of this technique and these measurements, the group in Cambridge, and the EPSSC in Windsor for funding. Thank you. And if you have any questions.
Thanks, Raj. Questions? Yeah, Chris.
Raj, within the region that you're probing with the beam, then am I right in thinking that there is actually a large assembly of the nanowire, of the...
Yeah, there's hundreds and thousands.
Okay, so you aren't able to, in a sense, just fabricate a single strand and then interrogate it?
We could. It depends on the size of the single strand that we would like to look at. So in this case, we looked at really sort of the really nanoscale systems, so like really small systems. But in theory, you could take a gold wire, you could take a... If you could
I was just wondering whether, because you then get some aggregate view of the transport, whether or not that's easy to understand, or whether or not you get
so it could be
that are more complicated and...
Yeah, it may be more complicated. They sort of maybe transfer between aggregates themselves, but the aggregates themselves are also very long, and they typically tend to orient themselves, and they don't really like, from sort of control experiments, we realize they don't really like to transfer energy between each other. They like to go in sort of one direction. It's very hard to get energy out of them, actually. So we're pretty confident that it's just aggregate transport.
Okay.
Time for one more question? Yep.
So a quick question. The type of transport we're looking at, do you think it's Dexter or Firster?
So I think it's neither, really. I think it's a sort of extended Firster type transport. But the timescale on which it's happening is sort of suggestive of some sort of delocalization of the wave function that's beyond a Firster type model. But I don't really know much more beyond that.
Okay, thank you.
No worries.
Okay, I think we should thank Raj again.